I am TANYA CHOWDHURY, and on behalf of NextGen Scientists Foundation, I welcome you all to this event. We at NextGen Scientists Foundation promote Indian life science students to pursue a career in science. And as a part of this initiative, we are running the NGS internship program, which offers financial support to Indian students who wish to undertake internship at any good laboratory in India. In an attempt at promoting science communication, we also have an annual essay writing competition. And since last year, we have built an online Discord based platform, the Indian Life Science Network, for encouraging seamless communication between life science students across the globe. Please do ask questions, press the raise hand button, and I shall put you on stage. Today, we are honored to have Dr. Dipyaman Ganguly with us from Indian Institute of Chemical Biology. Dr. Ganguly graduated in medicine from Medical College and Hospital in Kolkata, but then shifted his focus to biomedical research and joined Indian Institute of Genomics and Integrative Biology as a clinical associate. Later, he joined IICB for his doctoral research and earned his first doctoral degree. He next moved to USA and earned his second PhD from the very renowned University of Texas MD Anderson Cancer Center, followed by his postdoctoral work at the Columbia University Medical Center as an SLE Foundation Fellow. Presently, Dr. Ganguly is a principal scientist and associate professor A CSIR at IICB, and he has been making significant contributions to the field of immunology. A very warm welcome, Dr. Ganguly. We are really honored to have you with us today.
Thank you so much for having me here.
So, Dr. Ganguly, you started off with a MBBS degree, but then shifted your focus to research. What motivated you to deviate?
Well, I was very much interested in natural sciences. I was really decisive while taking up medicine that I should go for physics, honors, BSc, or medicine. But as you know, in India, the science students are pushed into all these professional courses. I took up medicine because I was also not bad in life sciences, and but after 2 years or so, I kind of decided that yes, I'll do medicine, but in a different way, because modern medicine, when you practice it, it essentially becomes maintenance of protocols, right?
Right.
Unless you are a surgeon. And if you are a surgeon, you are facing challenges every day, new challenges. Although you are following a protocol, but still there are unknown challenges. But I never wanted to be a surgeon. And then I decided, I need to find my niche while studying medical science or for MBBS, and then focus on that. That niche came upon me in the third year of my studying when we were studying immunology. Well, we were studying clinical immunology, and so the clinical immunology specifically focuses on the human immunology and the diseases that are related to immune system. And anyway, I kind of figured out that I wanted to do immunology research. I finished my MBBS, and then I was trying to get a break. And interestingly, in IGIB at that point of time, they were running a very big countrywide asthma genetics program, and they were looking for doctors. They wanted doctors because they wanted to stratify patients, recruit family members for the transmission studies. And so there were 3 doctors who joined that program. And so I think all three, some of you will be knowing all 3 of them. So one was myself, as I said, and the second was Dr. Ulganathan Mabali Rajan, who finished his
Oh,
MBBS from, I think, Coimbatore. And the third one was Dr. Sangeeta Goswami, who finished her MBBS from Guwahati Medical College. So Mabali and myself are in IICB currently as scientists, and Sangeeta Goswami is a very well-known cancer immunology researcher now. She's an, I think, associate professor in MD Anderson Cancer Center. She works with Pam Sharma and Chimalisa. Okay, so interestingly, one of my students, PhD students from IICB, now is doing postdoc with Sangeeta Goswami and Pam Sharma. So it's a very nice network of old friends. And so I started doing asthma genetics and started learning it. And when, in MBBS courses in India, at least when we took those courses, the research component or the introduction to modern technologies was meager. And I did not have any idea about how, what is the real doing research feels like. And that feeling was given to me in IGIT. And I started doing genetics. I soon figured out, yes, immunogenetics is not something that I'm interested in. I wanted to do cellular immunology. And then I talked to my mentor there. And I would say I was really fortunate to have great mentors all through my career. And usually, what happens in such cases? Your mentor will not let you go, right? So you have
Yes.
started one job and you should continue it and finish it, do your PhD from here, etc. But he was very sympathetic to my problem. So he was saying, okay, so if you feel like you want to deal with cellular immunology, then this is not the place for you. I'll figure out uh, where should you go. So he was again pointing me to go to USA at that point of time. At that point of time, it was really difficult for me for different reasons, financial reasons, etc., to go for USA right away. I already left clinics officially, and I was taking a lot of STEC, so it was a great time for nervous breakdowns. And so he suggested you should just then join an Indian cellular immunology lab, and I approached Dr. Shantu Bandyopadhyay and joined him. Interestingly, the work went really well in that lab. That was in IICB back in Kolkata, and I finished my PhD. But I figured out that although I am working on cellular immunology, I'm not being exposed to all the techniques or all the nuances of research question development and solving it while working in IICB. And as I already mentioned, I already took a great risk of leaving clinics. And I was not a bad student in medical science as well. And so all my friends were also really worried about me. So what this guy is doing? And so I thought that maybe I should train myself more, and I should really really get trained from a bigger place. And so I decided I'll go for another PhD, which will be a more focused immunology program. And that was so I was offered by Connecticut and MD Anderson. I chose MD Anderson and went there. Interestingly, I got to work with a guy who was really linked to uh, this dendritic cell biology field, which I am still continuing. So again, I was really fortunate. So Shantu Bandyopadhyay in IICB, so he was doing cellular immunology, but during his postdoc, he was one of the persons who actually discovered plasmacytoid dendritic cells. Well,
Okay.
that history is largely hidden. Um, and then there is an official discoverer of these cells as well, who worked in MD Anderson. So I got to work with that guy now. And so I worked with Michel Gillier and Yongjun Liu. So Yongjun Liu is credited to have discovered PDCs back in 1999. So I continued working on PDCs, plasmacytoid dendritic cells, and then finished my PhD in MD Anderson. That was quite a interesting time because so again finished our work was done, and after 3.5 years, myself and my mentor went back to the graduate administrator there, Stephanie. Stephanie was really worried to see us, what we wanted to tell her, and we told her that I want to graduate, and Michelle has no problem in letting me graduate. after 3.5 years, and MD Anderson was really aghast to learn that I'm finishing my PhD in 3.5 years, because they said that I was setting a wrong precedence. Anyway, my doctoral committee was populated by Michel himself, Yongjun Liu, and Chen Dong. Chen Dong, so he discovered Th17 cells and also partly follicular helper T cells. So these people
Okay.
were so big that when they suggested, no, this guy can graduate now, the graduate school kind of acquiesced to them, and I got my PhD and moved on to Columbia. Um, so this short PhD, what was good for me, that I did not really lose a lot of time, because my stay in IICB was for around 3 years, and in MD Anderson I stayed there for 3.5 years. So it was essentially 6.5 years. Then I went to Boris. Boris was working on autoimmune diseases, and again he's a big guy in PDCs because he discovered the master regulator transcription factor in PDC development, and he had this um, mouse which was PDC less. At that point
Mhm.
of time, he was the only lab that had this mouse. And I wanted to test some of our hypotheses that was generated during my graduation in his lab, and he was really welcoming, and I did that. So, and then I decided I will come back, started my independent case. I think you understand how the story goes.
Yes, absolutely. So, I mean, this is very interesting. I mean, what factors actually helped you in finishing your PhD so quickly compared to others, like where usually it's a 5 to 6 years or even longer time duration?
I think it depends on your projects. I mean, so if you're asking a very focused question, and if that question can be answered with few number of experiments, your PhD will be finished early, right? So it doesn't really. I mean, it's not. It's very stochastic, I would say. I mean, you can't imagine a project when you're starting it. How many experiments will help you to prove the point to a
Yes.
degree that it can be published, right? And
Yep.
so I would say I was fortunate, although I don't believe in luck or fortune, but I would say it was circumstantial positive vibe that helped me do it. Now, there is to some extent some aspects of it which is under control of a student. Uh, I think I have seen. I have not been in independent career for 15 years or 20 years, so I'm not saying that I'm a very experienced independent scientist. I've been doing independent research for 10 years almost now. I've seen most of the students are really focused. They really want to finish their PhD early. But at times, what happens, uh, the first few years, they tend not to get into the project too deeply or own the project. Once you start owning the project, the experimental schedule and experimental sequence becomes much more clearer to you. So then you don't have to depend on the mentor too much, and that actually helps to finish few things earlier. Because I have seen students not only in India, in the abroad as well, in my lab as well, where I did my PhD, where uh, they were not very sure about of the project's deliverables and what exactly they're going to uh, give the message in the paper that they are expecting to publish. So if once that message is really clear in the mind of the students that this is the thing that I am discovering. So my PhD mentor used to tell a very interesting point. So every publication should give one message, not a multitude of messages. Once you start giving multitude of messages in one publication, your project gets delayed. Your publication gets complicated. It becomes difficult to publish. So once you have zeroed in on one single message which is impactful enough to be worth of publication, and then prove it from the all possible dimensions that are within your reach, uh, the project ends up much earlier than it usually for other projects. I hope I could make you understand.
Yes, I understand. It's like what specific question we are asking rather than asking a multitude of different questions
Exactly.
at
So
the
I
same
think in
time.
most cases, even in projects that we run, at times we are lost in multitude of problems. We want to answer too many things, and that actually creates problems. We also suffer from those kind of things. As I said, I mean, this is little bit stochastic. At times, your brain clicks and you figure out, okay, so this is the direction you should take. You should just uh, stop other thinkings around the domain and just try to prove this point, and that will be impactful enough and will be um, good enough for a single publication. And then it works really fast. Uh, so this is not a personal quality. I would say these are again stochastic picking of brain, and that helps. But it the time can reduce if students really start owning the project really early and start thinking about it, because I have seen students look up to their mentor for a long period of time, too long, um,
Mm.
because they come from master's education and start PhD. But we have to, despite the dominant atmosphere in Indian educational institutions, uh, they should realize that in the masters there is a teacher-student relationship. When you are coming to PhD, it's more like a junior colleague and senior colleague relationship, right?
Yes,
Because the
yes,
question, because the mentor is mentor just because he has asked similar questions before and has successfully answered them before. But essentially, for the project that is you're starting, that question, I mean, he doesn't have any clue how to progress in that direction and what will be the answer, and that's how science progresses. The only reason a mentor is a mentor because of his or her experience of doing it before once, at least once. For students, this is the doing for the first time. But despite that fact, they are nothing but senior and junior colleagues. And once that kind of relationship is agreed upon, and both take equal responsibility and own the project, usually things go faster. Uh, but I mean, in Indian educational institutions, we still call our mentors as sir and madam and all these things. I mean, so naturally, this agreed upon junior senior relationship comes much later, and it depends a lot on the personality of the student, personality of the mentor, and so all the time it doesn't really work that well.
right. So, I mean, this is one aspect that you find is different abroad. I mean, based on your training that you have seen, that it's more like a colleague sort of thing.
Exactly, of course. So while you are working abroad, um, so they really look upon you. They really trust you for your intuition, for your scientific acumen while progressing in a project. In the lab meetings, they will actually look up to you, what you think should be done. But yeah, I think in India, I don't see that much. Usually, there is a one-way monologue and instructional uh, meetings, right? So that should not be the way it should be done. I mean, I feel so. There may be
Yes.
lacunae I don't understand. There are nuances, but definitely the junior senior colleague uh, dimension helps it a lot.
Right. So, I mean, then what do you think should be an addition to this in the form of a postdoctoral training that would help in sharpening the skills of a student?
I think you are now yourself a postdoc. I think you realize that in a postdoc, they are really looking for a junior colleague. They're not at all prepared. Most of the big labs where people try to do postdocs in, they are not at all looking at someone that she will be trained and she will be, then she will take up the project, etc. In all postdoctoral labs, I have seen that in good labs, the postdocs own the project completely. Now, the training for owning the project, if it doesn't start from the graduate days or doctoral studies, it becomes difficult. And so during postdoc, you are on yourself, and so you can actually now drive a project yourself. You can just seek for advice from your mentor at times, and that pushes you towards this independent career thing, right? So then you get some self confidence. Okay, so I can start one project myself and try to finish it, taking help from my junior colleagues. So that's the transition, right? So at some point of time. So I think the doctoral degree should be looked upon by mentors, by students, both the parties, as initiating the master students towards this junior colleague, senior colleague dimension and owning the project, thinking about it and finishing it together with the mentor and the student. And the postdoctoral thing should be the training for the postdoctoral uh, fellow of becoming the senior colleague now. And so playing the role of the senior colleague, at least to some extent. And then when you're independent career scientist, then you really become the senior colleague, and everything depends on you. So once you start looking at this from this point, I mean this perspective, uh, things becomes much easier. And also the often encountered tension vanishes. I mean, so uh, this is an ongoing process of training yourself to be the senior colleague who will own the project.
Yes, right. So you returned to India with the Ramanujan Re-entry Fellowship, and it's quite competitive. So how difficult it was to get the award, and what are the challenges that you faced while searching for host institutions in India? I mean, and also what were the hurdles that you had to face while setting up your laboratory for the first time?
So there are two questions here. First is looking for job and I mean getting a early career grant, etc. Ramanujan fellowship, uh, yes, it's competitive, but still it's not that competitive. Also, I mean compared to, for example, intermediate fellowship, Wellcome Trust, it's not that competitive, right? So Ramanujan is fine. I mean, once an institution decides that they want you or someone to be their future faculty, then only the Ramanujan Fellowship sponsorship comes. So I think that is more difficult. Getting Ramanujan Fellowship is less difficult than getting the sponsor for Ramanujan Fellowship. And naturally, so people who are already towards the end of their postdoctoral career, they should think uh, how to kind of position themselves or position their own CVs for different institutions. What people usually do, they apply to every place, I mean, all the places in India, because in most of the time in Indian institutions, at least when we came back, there was no focus or mandate in most of the institutions. But you have to remember these days, the institutions are trying to focus on few things. For example, I mean, I don't want to mention some of those, but you have to figure out from their list of faculties, the type of projects they're doing, that what kind of faculties they are trying to look at. If required, you can actually talk to their recruitment officials. I mean, usually one or two scientists are given the responsibility of recruiting new faculties. You can discuss. I mean, there is no harm in discussing with them what kind of people they are looking at. I mean, they are thinking about recruiting. And that will help you zero in on the institutions that you will approach formally. and that is very important. For example, if you do not have any teaching experience, you can't really right away go for university to jobs, right? Also,
Right.
ISARs have a lot of teaching responsibilities to take up. So they will always prefer someone who has some teaching experience, at least in terms of TA-ship during doctoral studies, things like that. So try to figure out what is in your CV that will entice that specific institution to take you up and sponsor you for these fellowships. Fellowships themselves, I don't think they are that competitive. Uh, I don't think they are. They have reached their ceiling of funding them because I mean every year I think 70 or 50 or 60 people get funded. If there are good candidates, I think they can go up there. But I don't know currently what will happen because very recently, I think all of you know, very recently there had been some government instructions of fellowships being dumped.
Mm-hmm.
Honestly, I don't have clear idea of which fellowships will be dumped because it doesn't really say clearly. I hope that they will come up with alternative avenues. If they are dumping few fellowships, hopefully they will create few new fellowships. Otherwise, it will be very difficult for people who are trying to come back. I don't have clear idea as of yet now.
Okay. So, coming to the next question, like, how difficult was it for you to set up a lab and how long it took?
That's a difficult question. It depends on the institution that you're getting in. For me, I wanted to be in Kolkata, so I had very little options. And because I was largely, I mean, the major motivation for me coming back was uh, taking care of my parents. And I was a lone child. And so naturally, Kolkata was the place I was looking for a job. And it depends on different institutions. For me, it took around 1 year in getting my own lab, and that to a very small corner lab kind of thing. But we started working from the very beginning because I was fortunate again to have very energetic initial students, at least two of them. And so I remember I started in a very small room with just sitting table and chair, and I did that for almost 8 months. And my I also had students, and they did not have any. I mean, I never had any lab at that point of time. And what I had was plans for experiments and what kind of instruments that will be required for those experiments. And I used to do my colleagues' lab, and and they were also very welcoming, I would say. That's how I actually started my independent work without having a lab. So that tells you that even if you are not having a lab, I mean, starting your lab, how long it took to start your lab can be answered or rather framed. You can frame the question in a different way. How fast you could start your work or project. So that depends a bit on you and your colleagues. If they are welcoming enough and you are pushy enough and you can pursue them, you can actually start pretty fast in any institution. But having your nice own corner of lab and having an office and all the instruments in your lab, it takes time. It takes time because of many reasons. There are institutions where there will be space already ready for you. I believe instruments will not be ready for you because here there are a lot of the financial regulations where your instrument you have to buy. Someone cannot buy you an instrument and give it to you, your lab, right? So I think
Yes.
people who have done PhD in India, they know it. Indians are here, etc. And buying one instrument in India is very expensive. I have a diametrically opposite example. For example, I visited China in 2015. One of my friend was appointed a professor there in Guangzhou. And I went there. There were 8 immunologists in the building. There were 3 floors with 27 labs. Only 8 labs were populated. They had 7 confocal microscopes, and I was wondering what they're doing with so many microscopes. I asked the head of the department, so you have 8 people. Why are you asking for? Why do you have so many instruments? His response was, "We are going to recruit 27 of them." Okay, so there are places where people think in a different way, but in India it's not doable. Even if the head of the institution or head of the department thinks that he will populate instruments and before the people come, it's very difficult to do it because of financial regulation. And so again, it depends on which institute you go and how things are. But having said that, as I said, if you are really trying to be in touch with your colleagues early on, before even you come, you start thinking about projects, how to do it, ask them, what instrument do you have, can I use it for few experiments. Most of them will say yes, and you can start right away.
Right. Great. So, all now moving to the focus of your research, like coming across your lab website, we found that you have been focusing on different aspects of innate immune system with a special emphasis on plasmacytoid dendritic cells and the systemic immune functions. So, what is the long-term research goal of your laboratory?
Okay, so our long-term research goal is, long-term research focus is on plasmacytoid disease. Okay, and
Mm-hmm.
so plasmacytoid disease, we know a little bit of it, but in terms of viral infections and their role in infectious biology. But their role in autoimmune diseases was actually part of my PhD work. And so at that point of time, 2008, 2009, people started realizing it, that these cells are really critical pathogenic cells in systemic autoimmune disease. Now, systemic autoimmune diseases include so many diseases: type 1 diabetes, SLE, psoriasis,
Right.
Sjogren's syndrome, scleroderma, rheumatoid arthritis. Interestingly, in most of the cases, this type 1 interferon production from these cells is very critical. So the major focus of our research is at one hand trying to find out molecular regulators in the PDC biology that can be targeted in different autoimmune diseases so that you can stop PDCs functioning aberrantly. And second, finding new areas of autoreactive inflammation where PDCs play a role. So in terms of the first dimension, we showed that yes, there are endocannabinoid receptors that regulate PDCs in some SLE patients. On the other hand, we found that Amrit did that work, found that in obesity-associated metaflammation, PDCs play a role. We were the first to show that. And so that's how we were working. Now, in 2015 onwards, I had a student in the lab with whom I started uh, like poking into this new field of mechanosensing in um, immune cells. And that question also came from PTCs. I can't discuss it right now because that story is still unpublished. So
Okay.
while we were working on PTCs, we found, we kind of got interested that there may be some mechanosensing in plasma membrane which regulates immune cell function in PTCs. And then we started looking into other cells, mostly T cells. We actually published T cell data on piezo-1 mechanosensors. The PDC data is yet to come out. That's why, so there is a very small group of, projects and students who are actually looking at this part of it, which is more fundamental basic immunology, to see whether membrane tension generation is part of immune function regulator.
Okay. So, why did you specifically focus on only plasmacytoid dendritic cells? I mean, there are different immune populations which also play an important role in mediating these autoimmune disorders.
Yeah, because these are the innate initiation events in these diseases. You know, so for example, in psoriasis, you'll find that it's the final tissue assault comes from Th17. In SLE, you'll find that the final assault comes from majorly autoreactive B cells, right, and the autoantibody deposition. So the end secondary immunocellular outcomes, I call them secondary immunocellular outcomes, and they cause the tissue damage. But the disease initiation comes from a very central event, and it seems ADCs are sitting at that center. And that's why we want to target these cells because that actually initiates the disease process and fuels it throughout the life of the patient. These diseases go on, I mean, it starts pretty early, usually in young female patients, and they suffer for almost rest of their lives. And only treatment is systemic immunomodulation through corticosteroids. Now there are some B cell depleting monoclonal antibodies, rituximab, etc., which is really highly expensive also, and it also doesn't really work in all the patients. And so
Mhm.
that tells you there is a need for new areas where these diseases can be targeted. And so that's why PDCs play a very central role. So they are the PDC activation, which was not due in a tissue space, is the initial event and the fueling event for the inflammation in these diseases, and we want to target them.
Okay. So, I mean, you always believe in a holobiotic view of the immune system, and
Right.
possibly it has inspired you a lot. So, could you shed some light on that? How do you feel that the overall physiological systems and parameters actually impact?
So my interest in holobiont, the concept of holobiont comes from a very different perspective because I personally am interested in very interested in the philosophy of immunology. So these are I personally do some of those work. So now immunology is a domain of life science where the application came before the subject was well known. So you know Edward Jenner, Lady Montagu, etc. So they came much before. So vaccination came much before we knew anything about immune system. And so, and also immune system, essentially when we think of immune system, we think that it discriminates against non-self and it protects the self. And that's how from the high school days to the medical school to through the doctoral studies we have been taught, or that's the major like dominant view, worldview for immunology. But if you think little deeply, you will find that there are a whole lot of clinical contexts where immune system doesn't really protect the self. I already talked about
Right.
autoimmunity. Now, this was first talked about talked by Polly Metzinger. I don't know whether you know this lady. So she was an amazing immunologist, largely theoretician. You know the infectious non-self model that infections provide pattern recognition receptor activators
Yes.
that causes immune activation, and that's how immune system senses non-self, and that was done by Charles Jenway, right? And from
Right.
his prediction, all the TLRs, toll-like receptors, all the receptors came up. But back in 2000, I mean early 2000s, Polly Metzinger proposed that immune system doesn't care about whether it's dealing with an infectious non-self or anything non-self. It actually cares about danger. Now, this danger, when you talk about danger, Metzinger was not well accustomed with microbiome research. Now, when you are doing immunology in 2022, after 20 almost 25 years after Metzinger initially proposed danger model, now you have to make sure you incorporate the data on microbiome and the concept of holobiont into the immune algorithm. Now, if you ask question, why is there an immune system in your body? The answer is very murky. You don't have an answer. So, along with one of my friend, and we actually uh, work together at times and also discusses. So he's a philosopher in Porto. He's and also uh, another experimentalist is in Porto who worked with me in Colombia, Vania Sisirak and Thomas Pradu. Thomas Pradu is a philosopher. So uh, we are trying to imagine a new model for immune system. I mean, it's very important to figure out when you have these loads of microbiome data where the infectious non-self is sitting right there inside your tissue, um, and there is no point in thinking about danger because danger is within your household, right, in the body. So how
Right.
exactly do you figure out that the system works? So we are not very sure. And that's where my interest in holobiome comes from. It doesn't really only come from the microbiome research in different autoimmune diseases. We do that kind of things just to understand
Mhm.
how immune system responds to it. But I think there are bigger questions left unanswered, and we need to answer them.
Yes, so coming to the microbiome thing that you just now mentioned. So, some of the organisms are well tolerated in our system compared to the pathogenic ones. And it is the normal microflora that actually helps in keeping the pathogens away. So, I mean, do you feel it's a difference in the PAMPs or the patterns or the DAMPs, whatever we call it by the name, that actually helps in initiating this response? Like which one to consider as our own and which one not to?
I don't think so. I mean, well, there are people who thinks in this manner, but I don't think so. I personally think that you have to remember these organisms, you are discovering them today, but they were there right from the beginning of Homo sapiens physiology, right? And
Right.
that's 300,000 years ago. Now, this is that means your Homo sapiens physiology has been, I mean, Homo sapiens or any other multicellular organism, they're not really multicellular organisms. We knew them to be multicellular till like 2 decades back, but they are actually polyspecies multicellularity. And they have been so from the very beginning. Naturally, when you think of a multicellular organism as a polyspecies organism, the physiology must depend on each other. So uh, so our immune system was developed like that. Our immune system depends on its minor species inhabitants. For example, there have been very interesting data showing that segmentus filamentous bacilli in gut, they drive Th17 response. So if they are not there, those mice are not having a lot of Th17 cells. That tells you that immune system and the cell subset differentiation, division of labor, they are very much dependent or very much regulated by the minor species inhabitants in your body. And naturally, it's nothing to, I mean, it's not only just differential PAMP expression, it's much more deeper. It's like a co-evolution of minor species versus major species. I'm talking about the multicellular organism as a major species, which is the biggest one in this holobiont, and all other multicellular and unicellular organisms inhabiting within that body space as minor species. So I think minor species regulate major species physiology to a great extent, and we are discovering them now, but they have been doing so from the very beginning. So it's much more deeper relation.
So you feel it's more like a symbiotic relationship
Exactly.
where one is
Exactly.
okay. Yes. So basically, the signaling, some signaling cues and molecules from these organisms or microflora that we have, they are helping in shaping our immunity.
Exactly so. Exactly so. It's
So, like,
too network. I mean, too complex a network. So we are figuring it out. I mean, we are yet to figure it out completely. But the emerging data suggests that not only immune system, a whole lot of other systems also, but more true for immune systems. That shaping the immune system, the division of labor of cellular subsets, specific cellular subsets to be there or not, so they are all regulated by minor species metabolites, perhaps.
Yes, that is true. So, I mean, yeah, that's absolutely possible that you mentioned that the signaling cues are actually. So, let's say if we have certain transplant experiments where we put in the healthy microbiome of a immunologically fit person to another person who is having an aberrant immune system or maybe a very weak immune system, do you think it would improve?
I mean, there people have already done such experiments. They are doing such experiments. I think people have already shown that transplantation, like graft rejection activities, can be improved by microbiome, like fecal transfer or something um, from different organisms. So people have been doing those. In fact, in case of transplantation, this will be much less applicable, even though there are data showing that. But you will realize, I mean, in Parkinson's disease, people have shown that if you take microbiota from a Parkinson's disease fecal material and give it to a mouse, the mouse develops motor abnormalities. Just think about it. And so this is, I think, we are just we are excited about the effect of microbiota inside our body, but I think we should be excited about the finding that we are polyspecies organisms. We are not monospecies organisms. I think that's the major exciting part of it.
Yes, absolutely. So if anyone has any question, please raise hand and I would put you on stage so that you can interact directly with Dr. Ganguly. Uh, okay, so moving to the next part, it's like the COVID pandemic has been very challenging for each one of us, but you have been actively participating in COVID research during that time, and it has led to multiple high quality work. So, can you briefly tell us about your experience, the technical challenges and hurdles you faced while you were collecting the samples and conducting the research?
Well, I describe our work in COVID as response to pandemic. Yeah, we were responding to the pandemic, and I thought at that point of time that if there is a pandemic that is creating such a havoc worldwide, and if you are an immunologist, you should find out ways to contribute, because otherwise there is no meaning of the scientific enterprise. If in such
Exactly.
a dire situation, a scientist, he or she may have other research foci for rest of her career, that's fine. But when you are facing such a dire situation and you are an expert in a field, you should find out ways to contribute, and that was the major motivation that I worked on it. And Council of Scientific and Industrial Research was, so I was part of a small group where discussions on immunology was being held and what can be done, how things can be improved, and these were really early days. I mean, just after WHO declared it as a pandemic, even before the actually those meetings started. And there was, I mean, thought of running a convalescent plasma trial, like in all other pandemics. It has been done, and we started this convalescent plasma trial in Kolkata. As you can imagine, in India, it becomes little difficult if for scientific organizations to do it, because we are not associated with hospitals,
Yes.
and perhaps because of my association with different hospitals and my clinical background, they thought that I should do it, maybe, which actually helped, because I knew a lot of, well, all my medical school days friends are now practicing doctors, so I could go up to them, and they also we are in touch, and so that was the major reason I was perhaps commissioned for this research, and it also gave me the opportunity of contributing in that dire situation, right? And our, I mean, for the first few months, it was lockdown, and the institute was closed, and for a practicing scientist, it pains when their labs are closed, and it
Yes.
actually it actually helped me work during that lockdown as well. I think that was also another motivation that I'm still working. And yes, we did a fairly small single center randomized controlled trial. We performed the randomized controlled trial in a very good way, because I would say, because the convalescent plasma were well characterized, and blinding of randomization was very strict and all these things. I would this I must say that the much bigger multicentric trials done in India were not that stringently performed. Um, some other day we can discuss about that, but we actually took we take care of it in a very stringent way, and um, we delivered whatever data we got. And now I think you will be interested to when it should be interesting for you. You know, the WHO already banned convalescent plasma usage.
Yes. Yes.
And so in our trial, there was a precision medicine aspect of it, right? So there were few patients who were responding well to convalescent plasma. And um, Arturo Casadeval, who is the head of the public health and microbiology department in Johns Hopkins. So he noted our trial, and now, based on our trial and also a lot of meta-analysis done in different parts of the world, WHO has been sent a letter, a multinational letter, asking for lifting that ban, because you never know when another new variant comes where all the existing antibodies will not work. Even the currently circulating BA5 and BA4 Omicron variants, they are not amenable to any of the monoclonal antibodies, and and also the vaccine induced neutralizing antibodies. So we are saved because they are not causing very severe disease, perhaps because of some other antibodies, for example, the more structural protein antibodies. But you never know. So now uh, I would say that based on some of our work, uh, there has been this realization that in some patients CPT actually worked, and so there has been an application to WHO to lift that ban. If some of you are interested, I can share that document as.
Absolutely. And like, what is the key finding of your research? I mean, the entire duration of time that you invested in on this COVID research, what is the major finding?
So I think the major finding that we showed, that we went into convalescent plasma thinking of neutralizing antibody provision, right? Passive provision of neutralizing antibody to the recipient. But our data showed that when a person is recovering from the infection, there is a very interesting anti-inflammatory proteome that builds up in its circulating blood. Uh,
Mm-hmm.
there have been some research in this field, but inflammation resolution is largely unattended in immunology and inflammation research, because usually at that point the patient is not having any clinical issues, right? They are not clinically symptomatic. We
Yes.
figured out while working on convalescent plasmas and doing the proteomics analysis in greater depth, that there is a very active inflammation resolution proteome, and that comes up in convalescent plasma. And when you actually transfer this to an active patient, this inflammatory proteome helps them as well. And that was, I think, a very interesting finding. We never thought we are going towards that, but we actually found that yes, you can segregate the effect of neutralizing antibody and the anti-inflammatory proteome in the convalescent plasma, which also plays a big role in recovering patients.
But is it true for all patients? I mean, is there any variability in this phenomena that you have observed?
So there are variability in responses. I think so. It would help with the data, but there is variability of responses. You will find that the person is getting similar amount of anti-inflammatory proteome, similar amount of neutralizing antibodies, still not recovering. And we found that there is a big effect of the disease course, the duration of the disease course, and the age of the patient. And so that is really intuitive, right? Because aged individual, there has been in other clinical contexts there have been a big body of literature of aging lung. I mean, how the lung fails to respond to therapy in aging individual, and we feel that similar things happen there also. And now the evidence-based medicine usually averages out, so it makes an average of the cohort, right? And that based on that, it negates or posits use of such certain drug. But in the precision medicine domain, uh, the major focus is on finding out the subgroups in your cohort which actually respond to a drug. Because if in a disease there is no drug, and there are certain subgroups which actually respond to one drug, but when you average out on a bigger cohort, it goes away because there are some other patients who do not really respond to that drug. You miss out on saving few patients, and that's how precision medicine comes up. And I think ours was the only effort on that precision medicine focus in a CPT randomized control trial, and that was the uniqueness. And I believe in most other clinical contexts this works. Even you'll find, so you use paracetamol for getting rid of fevers. We don't really know. You should remember, we don't know how paracetamol reduces fever, and we have been using it for so long. Uh, Keeping aside, so you'll also find there are individuals who needs uh, higher dose of paracetamol to get rid of fever. Paracetamol, I gave
Right.
the example of paracetamol because this is the most homogeneously responsive therapy. Fever, uh, assuages, it actually, you can get rid of fever by paracetamol usage. But still there are variability of response. So now you think of other kind of drugs and how variability of response will actually affect their therapeutic outcomes. And that being said, I'm just trying to point out that when you are in an evidence-based medicine, when you are doing randomized controlled trial, you miss out on certain subsets of patients who might have responded to your therapy being tried. So these days, that's why in the pre-specification of clinical trials, people are going for subplus analysis. And this is a good trend, I would say. This is what we should go for. Because that will tell you that if there are patients who will be nicely responsive to a therapy, and there will be patients who will be not that responsive. And in that way, our options increases in terms of developing new therapies.
Yes, absolutely correct. So, with respect to the use of recombinant antibodies specifically directed against neutralizing this COVID infection, do you feel that covalent plasma therapy is better option than that one?
Of course not. Convalescent plasma is an emergency response. So monoclonal antibodies are the final product, right? So how do you generate these monoclonal antibodies? You generate from convalescent individuals' B cells. And when you are
Yes.
facing a pandemic, you are not having that product. And definitely those are the best because you are giving neutralizing antibodies. So if you give a neutralizing antibody early enough, it should prevent severe disease from happening. My point is different. My point is that convalescent plasma will be required now if tomorrow there is a variant which is not amenable to any of the existing monoclonal antibodies, and again you will need 6 months to 1 year to develop new set of monoclonal antibodies, right? And so that's how the use of convalescent plasma is always sought for, to give an interim management.
Okay. Yes, that's an interesting finding actually from the group, and it has been really highlighted also. So So, as an immunologist, what do you feel is the scope of immunology research in India, and what are the challenges, or which areas should India specifically harp on?
So I think immunology community in India is really small, uh, really small. I frequently say this in different meetings that the number of uh, real immunologists in India is less than the number of immunologists that are in the Manhattan Island, and still it's so. I mean, I think the first time I said this when I first came back here and attended one Indian Immunology Society meeting, and 10 years after that first meeting, still I think it's the same. So we need to have a large number of immunology labs because immunology is going to be the future of uh, medical science. Because now we are slowly understanding how immunology can be harnessed for developing therapies in different clinical contexts, in contexts where people never thought you can actually use immunology. And so naturally, we need to have more immunology labs. We should have more immunologists working in infections, definitely, but also in all the chronic inflammatory contexts, starting from autoimmune diseases to metabolic syndrome, cancer. If you think about it, in India, how many cancer immunology labs do you find? It's such a big country.
Yes.
Very few. How many immunology labs work on SLE? Very few. I mean, for example, PDCs. I think in the whole India, myself and Praful Taylor works on PDCs. Praful Taylor is a very amazing scientist in NII. So that's the way it is. While if you go to just Manhattan Island, how many people work on PDCs? I think you will find like seven of them. So that's the difference we have. So we need to have more immunologists. I hope so in the PhD entrances of IICB, I have seen in also in few other institutions outside. I have seen most of the people come and talk that I want to work on cancer biology or molecular biology or something like that. I know they are not very, they are not initiated to the field, but I hope that very soon they will come and tell us that I want to work on cancer immunology or I want to work on autoimmune diseases. Uh, so that is very much important. That will actually generate more immunologists because Indian immunology community will be fed by Indian immunologists. And if Indian students are not taking interest in immunology, how can you get Indian immunologists? I don't think an American immunologist will come and start a lab in India, right? So I
Yes.
wish a lot of life science students take interest in immunology because immunology is going to be big area of life science research and more prolific. I mean, new discoveries will be made, new therapeutic avenues will be opened up through immunological research. And so I think a major chunk of life science students should take interest in immunology.
So, are there specific government agencies or bodies that are eager to promote research in immunology in India?
Well, I believe DBT is thinking about it because DBT started immunotherapies. I mean, there is a committee on new drug development and vaccine development and immunotherapy development. So that tells you that they are really realizing that immunotherapies are separate domain, exclusive domain, and you need to specifically promote and nurture those labs in the country. Uh, Once you find a whole lot of immunology labs starting in India, definitely there will be more focused nurturing efforts because you don't really have a lot of labs. Uh, very few of them.
Yes. So, now moving outside of academia, like you are a senior scientist, a teacher, a father. So, what do you feel how a person should manage to have a very healthy work life balance?
Well, it may sound a little controversial, but I think if you love your work, if you love what you do, this confusion about work-life balance doesn't come. Um,
Yes,
I don't know
I
whether you agree with me
I
or
agree
not.
with you. Yes, I absolutely agree with you on this point. But several students in their initial stages, they do have a lot of questions about this. But I absolutely agree with you that if it's like what you really want to do, then it no longer stays as a work.
Exactly. So I think the major problem comes from students joining PhD without thinking much about it. So they come to do PhD, a lot of them. I mean, I'm not saying all of them. A lot of them, they come to do PhD as a reflex response after doing masters in certain field. But that's not the way it should be done. I think if you're really feeling for doing research, because don't don't aggrandize this profession. There is no need for aggrandizing this profession. That this becoming a scientist is a great thing. Becoming a teacher is not that great. No, it should suit your personality. Unless you are badly feeling for it and you want to do research, I mean, then only you come to do research. I mean, otherwise, uh, this is specifically true. I mean, more true for research career because here there will be a lot more ups and downs compared to any other profession. But you should remember that we are also the luckiest bunch because we are, at least in India, we are given salary from, we get our research expenses from public money. And public largely doesn't have any inkling about what we do. They don't ask us questions. Just think about a doctor. They are being asked so many questions these days in India. And for scientists, we are luckiest bunch, right? So they don't really ask a lot of questions. So we motivate ourselves. We have to be a little responsible because you are dealing with public money, and also you need to understand why do the research money comes from the um, fiscal commitment. It comes, I believe, I want to believe in this way. It comes from the fiscal commitment of government just to continue the legacy of scientific enterprise in the country. How do you continue the legacy of scientific enterprise? By generating more scientists. And not only generating more scientists, by generating better and better and better scientists. For example, if my student doesn't surpass my achievements and becomes an Indian scientist after 20 years, because after 20 years I'll die, and there should be new scientists, and that's how the scientific enterprise in the country will continue. And so my responsibility is to generate new generation scientists, like your foundation is named Next Generation Scientists. That's why I was saying that it's a very interesting name, and so our fiscal commitment is generating next generation scientists. And our fiscal commitment is also to make better next generation scientists. Because if we are not making better next generation scientists, the growth of science in this country will remain stagnant. If you want the science to progress, progress means there will be an upward curve. That means my next generation has to be better accomplished than myself. I don't think we think in this way when we join PhD. I mean, so PhD is just another profession. People who are really feeling for it, who really think that unless I do scientific research and do experiments on my hand, I can't get a good sleep at night, they should come to research. Intellectual brilliance doesn't matter. What matters is motivation and enjoying what you do.
Yes, I absolutely agree on this point with Dr. Ganguly. You really have to be motivated to be in research. That is the only driving force that will keep you alive. So, do we have any questions from anyone? Okay. If not, no issues. Thank you, Dr. Ganguly, for joining us today. It was a pleasure to have you with us, and it was a wonderful discussion. I hope we'll have a few more with you eventually on some separate topic. But thank you so much.
Thank you so much. Thanks for having me. Best wishes to all of you. Thanks.
Thank you. Thank you all, and good night.